The CDR3 Well Plate Explorer allows the user to display TCR BCR data in a well plate format within grids. There are four main data display options which can be selected in the top left panel. CDR3 by plates and wells displays the data within the primary grid, ordered by each plate and well. For this data, each well contains a single cell, and the well should be made up of one or more rows consisting of A for alpha and B for the beta chain. There may also be a D delta and G gamma chain. If this was B cell data, then here we would have the H for heavy and K or L for kappa lambda light chains. Therefore, the cells in the first well A1, shown in grey, has two rows consisting of the TCR's alpha beta chain. Double clicking on the first column displays all unique CDR3s in the secondary grid. Here we have the number of times the CDR3 is seen across the plates and wells, and the max, min and average clone count. We then have 5 columns where the numbers represents the various CDR3 well combinations. To see a description of these combinations, just click on the question mark in the column header. So the first one is the number of times the CDR3 is present. So if we click on the cell, we see that this CDR3 is present 125 times, which is then displayed in the secondary grid. The second column is the number of times this matching CDR3 combination is present. So we see this combination 56 times. The third one, the number of times the CDR3 is present in a combination which does not match this combination. So we see this CDR3 22 times in a combination which does not match this combination. The fourth one, the number of times this CDR3 is present in a combination which does not contain any other CDR3s present in this combination. So we see this CDR3 12 times in a combination which does not contain this CDR3. The fifth column of numbers represents how many times this CDR3 is seen with the other CDR3s in the well, regardless of any other CDR3s in this well. The numbers are ordered from the first to the last CDR3 in the well, with the current CDR3 row missing. Therefore, the first CDR3 is seen once with the second CDR3, 66 times with the third, once with the fourth, and 3 times with the last one. The second CDR3 is seen once with the first, once with the third, once with the fourth, and once with the fifth. Then we have the plate and well info and the clone counts, etc. Then the V variable and J joining gene segments for the alpha or beta chains. If a / symbol is seen, then this couldn't be identified and was either picked up as a 12 or an 18. If an # symbol is seen at the end, for example 12 #, then this could have been identified as a 12-1 or 12-2, etc. Within the data display option panel, we also have a clone count cutoff setting which updates all the data and the grids to only include results with clone counts above or below this cutoff. Initially, the cutoff is set to display everything, i.e. all results greater or equal to the minimum cutoff. Here too. The clone count track bar can be scrolled from the minimum to the maximum observed cutoff with the value being updated in the box. Alternatively, the cutoff value can be entered into the box followed by the enter key. The less than or greater than checkbox also allows you to view results above or below the cutoff. The second display option, CDR3 combinations, display each CDR3 combination along with the total combination counts regardless of plate and well. These columns represent the same combination types as in the previous grid. Again, each description can be displayed by clicking on the column headers and can be double clicked to view the data. The third option is alpha-beta pairs. For T-cell analysis we have alpha-beta pairing, or for B-cell then we would have heavy-light chain pairing. Here we list the CDR3s for all possible alpha-beta pairings. By double clicking on the CDR3 column, the secondary grid lists all occurrences of the CDR3. 12 is the number of times these pairs have been seen together within the same well. If we double click on this cell, then the secondary grid lists all CDR3s ordered by clone count within each one of these wells. The green AB identifies the CDR3 from the primary grid. We also have the question marks in the column headers to show a description of the columns. Here, the clone counts for both the alpha and beta CDR3s within the same well, ordered by clone counts alpha plus beta. Red indicates that this is not the alpha or beta with the highest clone count within the well. Here we can see that there's also another alpha within this well with a clone count of 168. We also have 3 optional checkboxes within the data display panel. The question mark gives a description of these. So we'll cut down the data by setting the clone count to 500. If we check the first one, the grid now includes the listing of all single non-alpha beta pairing single CDR3s. So here we see a beta 3 times within a well with no alpha pairing. The second check displays extra columns listing all times that either this alpha or beta was seen in a well but not paired together with the alpha and beta. So this beta was seen 59 times not paired with this alpha. Again, the green indicates that this is the alpha or beta with the highest clone count, or red with the lowest. The greyed out clone count means that there is no pairing present within this well. No pairing. Pairing. Please note that these columns, the 2 rows, represent the same well. However, for these columns they do not. They're not paired. The final tick just displays an extra row showing the alpha or beta's highest clone count within that well if the CDR3 did not have the highest clone count, i.e. it's shown in red. The V and J genes and the count per plate is also shown. If more than one V or J gene was present for the single CDR3, then this would be highlighted by colouring the second cell in red. CDR3 plate combinations. This lists how many times each CDR3 is seen within a plate. By double clicking on the CDR3 column, the secondary grid lists all occurrences of this CDR3. The plates column lists the plates along with the plate count within the brackets. Plate 1 30 times. Plate 2 29 times. The last columns represent each plate listing the count. This helps to easily visualize which plates the CDR3s are seen. These can be ordered in both descending and ascending order by clicking on the column header. Finally, the checkbox also displays a max, min and average clone count column next to each plate clone count column. So within plate 1, this CDR3 is seen 40 times, having a maximum clone count of 6474, a minimum of 72 and an average of 1549. All the data for each grid can be exported to Excel spreadsheet. All the data displayed in all 4 primary grids are saved in a worksheet, plus also the data currently displayed in the secondary grid, if any.